Hello everyone, welcome back to a new tutorial. This tutorial is about the control of gene expression. The entire genome of an organism is present in every cell that contains a nucleus. This includes genes not required by the cell. So, the expression of genes and the rate of synthesis of protein products like enzymes and hormones has to be regulated. So, genes can be turned on or off, and the rate of product synthesis can be increased or decreased depending on the demand of the body. This happens by controlling the genes at different levels of gene expression. In the previous tutorial, we have studied that gene expression occurs in two processes or in two steps. The first step is called transcription, and the second step is called translation. So we are now going to see how can genes be regulated before and after these processes. The first way of regulating the genes is the transcriptional control, which actually happens at the transcriptional level before transcription takes place. Transcriptional control can happen by histone modification. As we know, the DNA coils around histones because they are positively charged, and DNA is negatively charged. So, histones can be modified to increase or decrease the degree of packing or condensation. As a result, the more the DNA will be coiled around the histones, the less the genes will be transcribed. The transcription of genes is not possible when the DNA is tightly wound because RNA polymerase cannot access the genes. Another regulation that happens at the transcriptional level is the lac operon. The lac operon is a group of three genes, LAC-Z, LAC-Y, and LAC-A, involved in the metabolism of lactose. These genes are regulated by a repressor protein that prevents the transcriptional of these genes in the absence of lactose. So, when the lactose, which is also called an inducer, is absent, these genes cannot be transcribed. Whereas, when the lactose is present, or the inducer, it binds to the repressor protein and hence transcription happens. Regulation of genes can also happen at the post-transcriptional or pre-translational level, which actually happens after transcription and before translation starts. And these modifications actually happen to the messenger RNA, which was produced from the transcription. During the post-transcriptional control, a cap, which is a modified nucleotide, is added to the 5' end, and a tail, which is a long chain of adenine nucleotides, is added to the 3rd end. These both help to stabilize the messenger RNA and delay its degradation in the cytoplasm. Finally, regulation can also happen at the post-translational level, and this is called the post-translational control, which happens after the translation takes place. This actually includes the following: the addition of a non-protein groups such as carbohydrates chains or lipids or phosphates. Moreover, the amino acids can be modified and bonds such as the disulfide bridges can be formed. And finally, the proteins can be folded or shortened.